Good morning, everyone, and welcome to the fourth day of our online program for the Introduction to Microbiology and Biotechnology, organized by the Department of Microbiology and Biotechnology, Sister Nivedita University, Kolkata. For the last 3 days, we have done a very beautiful journey through cell to biomolecules and through the molecular biological methods and processes. So today is another day of an excitement, and we have two very interesting talks. So I hope that you all will enjoy today's lectures also. And on behalf of the Department of Microbiology and Biotechnology, Sustainability University, I, Rajat Pal, am inviting and welcoming you all for today's session. Good morning to everyone. Hello, hello.
Yes, Ajita.
Yes. So maybe I was got
Yeah.
disconnected. Yeah.
No, no.
So
You're
okay. So welcome you all for the fourth day of online course on Introduction to Microbiology and Biotechnology, organized by the Department of Microbiology and Biotechnology, Sister Nivedita University. On behalf of this department, I, Rajat am welcoming you all for this day. For the last 3 days, you have made a journey with the different aspects or different avenues of microbiology and biotechnology. Today, we are going to have two very interesting talks. And may I know, Professor Ghosh, whether she has joined or not? Dr. Ghosh, please. Dr. Atreyi Ghosh.
Yes, I have joined, sir.
Yes, yes. Okay, okay. And Mr. Ghosh, uh, uh, is he joined so that
Yes,
I can?
he will be joining just 15 minutes before his talk. He has already informed.
Okay, okay, okay. So he has to join by this link. Okay, so by which we all have joined. So just kindly inform him, and let's start for today's session. This is my immense pleasure to introduce one of our eminent faculty of the Department of Microbiology and Biotechnology, Professor Dr. Atreyi Ghosh. She is a tremendously good researcher, and she has, within the very, small time, she has done a tremendous job in the research. She has, put a tremendous contribution to the departmental research, and she has given an extra level to the research in the department. Till now, she has collaborated with so many, universities in India, and I'm very much happy to share with you all that already she is in a process to collaborate with the foreign universities as well. She has done her graduation in botany from Presidency College, Calcutta, and she has done her master's from Kalyan University, and after that she has done her PhD doctoral degree from IIT Guwahati, and she has a postdoctoral research also from Yenipoya University, Bangalore. After that, she has joined as an assistant professor in the Department of Microbiology and Biotechnology, Sister Nivedita University. So today she is going to talk about the bacteria and virus. The title of her talk is Bacteria and Virus, the Unseen World. So actually, we are going to see through her eyes today what the unseen world is, what we cannot see, that she will let us see today. So I am very glad to introduce and invite Professor Atreyi Ghosh. And madam, please take the screen. Today, in this virtual platform, I cannot say that please take the charge of the stage. So now everything is on screen. So please kindly take the screen. The screen is yours, and kindly go ahead with your very enlightening lecture. Professor Atreyi Ghosh.
Yes, so Dr. Roger Pal, he's our head of the department, and I am overwhelmed for this introduction. And first of all, I would like to know, am I visible to all and am I audible to all?
You are audible, madam, but you are not visible. At least I cannot see your video.
But my video is on. I don't know whether.
STC, sir, can you see AG, madam?
Rajatha, ma'am is visible. Uh,
Okay, okay.
I am requesting you to go to the toggle tile view that is present in the right side of the screen, at the right side at the downward. There, by clicking that, you can see the speaker. That is Atragos.
Okay, okay, okay.
Okay, okay. Yes. Yes.
Okay, yes, I can see myself and I can see many other students and participants have already joined. So, again, I am very much overwhelmed with the introduction. I have never had an introduction of myself before. So, first of all, I would like to thank to my department, microbiology and biotechnology, to all my uh, fellow faculties and all my students, all my dear students, and I am grateful, I am blessed to have our Honorable Vice Chancellor Sir, Dr. Dhruv Chattopadhyay, and also Dr. Ashok Ranjan Thakur, who always, Thakur Sir, to always be with us and guiding us with every challenges we are facing every day with our researches, and we the faculties and the students are very much motivated to take our department to a different level altogether. So I'm a very fortunate one to have a part of this, to be a part of this department, and I thank to all the participants listening to me in this talk and all the participants who have joined the previous lectures, the previous three lectures, and we are getting a very good support that is visible in our Facebook account and all the social media. So I'm thankful for everything. So let's start with our lecture today. Uh, so I'm going to present my screen over here. Uh, Shomshubra Thakur sir, please help me out with this. I am sharing the screen. Is it visible now?
Yes, it is visible now.
Okay. Okay. Bacteria and viruses, the
Ma'am, I want to request
this,
one. Okay. Now it is full screen. It is okay, ma'am.
yes. Is it visible?
Clearly, yes, it is clearly visible, ma'am.
Okay, thank you, sir. So, I would like to start my lecture with the contribution of great scientists, those who started the journey of microbiology. They were the scientists long ago, like 400, 300 years ago. They have started their journey with a curiosity and with a devotion, because nowadays we know that the government of our country and all the countries, most of the countries are giving fellowships for scientific researches. But in their time, it was not possible. It was not so uh, famous or not so encouraging by the government. So they used to do their experiments for their own curiosity, for their own knowledge. And their devotion, their love for science is unparalleled compared to our time. So first, I would like to talk about the father of microbiology, Anthony von Leeuwenhoek. I hope most of you know his name. He was born in 1632. He was a Dutch scientist from Netherlands, and he's known as the father of microbiology because he invented the first simple microscope to see the microbial world. Okay. Next picture is of Edward Jenner. He was born in 1749, a long back. He was a physician and scientist, and he actually invented vaccine. Actually, he discovered the concept of vaccination, and all the discoveries were by chance, and it was not planned, but it was accidental, and it was for the great welfare of the human being. Then Louis Pasteur, he was born in 1822. He was a French biologist and chemist and discovered the principle of fermentation and pasteurization. Now, without him, without his contribution, present day dairy industry or any industry like brewery industry, beverage industry, food industry was impossible. And also, he modified or he uh, constructed the concept of sterilized culture. Next comes Robert Koch. He was born in 1843 and he won the Nobel Prize in Physiology and Medicine in 1905. And the bacteriology term was coined by him, and he formulated the infectious pathogens which are causing infectious diseases, and he uh, kind of modified in a very concrete way the whole world of bacteria, which are human pathogens. Nowadays, whichever bacteria is known for their pathogenesis, the discovery of those bacteria started by holding the hands of Robert Koch. After that, Bejering came. He was born in 1851. He was a Dutch microbiologist and botanist. He was founder of the term virology and environmental microbiology. He was a botanist and he founded that there are microscopic world which is beyond the reach of microscopes. We need electron microscopes to see them. They are the viruses which are in between bacteria and in between the living and the non-living world. Then came Frederick Griffith. He was born in 1877 and he discovered or he gave the concept of bacterial transformation, which is the basis of modern day recombinant DNA technology. Without his findings, without his discovery, modern day biotechnology was impossible. And I have to talk about Sir Alexander Fleming. I hope you all know about him. He was born in 1881 and he was a devoted scientist from Scotland. He was a physician and microbiologist, and he also won Nobel Prize for Physiology and Medicine in the year of 1945. And we all know his contribution. He discovered the first effective antibiotic from penicillium. Then I have to talk about two scientists who jointly got the Nobel Prize in 1986, Ruska and Null, or we normally call Null and Ruska. They invented the electron microscope, without which the many of the stories of microorganisms, like viruses and all, were incomplete. They were mechanical engineer and physicists who came to contribute in the field of biology. So, coming to the world of microbiology. Now, if I talk about bacteria and viruses, they are the member of huge cosmos or huge microbiota, uh, which can be distributed in three groups, that is bacteria, archaea, and eukaryota. They are forming the world of microbes. Okay. Now, the organisms which cannot be seen with our naked eye, we need to have a microscope for that. That is why we call them microbiological organisms or microbes. Now, these microbes can be distributed in three groups: bacteria, archaea, and eukaryota. But if you talk about viruses, they are non living and living as well. They are just in between living and non living. That is why they are not considered into the living world of the microbes. So they are excluded. Okay. Now, the microbiological world, if you see, the archaea that is shown in the middle of the slide, archaea came first in this earth. They are the most primitive members of microbiological organisms. Then came the bacteria and the eukaryota. Now, bacteria and archaea, they both are prokaryotes, and the eukaryota, they are eukaryotes. Now, what is the difference between prokaryote and eukaryote? I hope many of you know, but I would like to just repeat it that uh, prokaryotes, they do not have the nuclear membrane in the cell, and the eukaryotes, they do have a nuclear membrane in the cell. And the eukaryotes, they have membrane bound organelles. Their cellular structures are a bit complicated or complex compared to prokaryotes. Now, the prokaryotes can be grouped into bacteria and archaea. Archaea are the primitive one, and the bacteria came after archaea. Okay. Now, this is a diagram which shows the evolutionary relationship as well. The different types of bacteria, how they have evolved, the branches which are more close to the source, they have evolved before the bacteria or the organisms which are placed at the upper side of the branches.
Excuse me, ma'am.
Yes, anybody is having a question, we can take the questions after the presentation.
So, ma'am, sorry to disturb, but can you just hide the option of screen sharing?
Yes,
Can you just hide the option of screen sharing because it is somehow hiding the line which is written at
Yes,
the bottom?
AP madam. Yes, yes. Plus, yeah. Now it is
yes,
fine.
yes. Thank you, thank you, thank you. Okay. So there are some differences between bacteria, archaea, and eukaryotes. Like I have said, the main difference is in their cell wall. So in the cell wall, the bacteria, they have a compound which is known as peptidoglycan, which is present in bacteria, but they are absent in both archaea and eukaryotes. Okay. Now there are other differences as well in the terms of membrane lipids, RNA polymerase, and other points are there. So I would like to talk about the other classifications of bacteria. So the bacterial world can be classified into many groups based on different categories. First is the nutritional requirement of the bacteria. Now, the bacteria, they are all unicellular prokaryotes. They are all unicellular prokaryotes, and they carry out their metabolism in various manner. They take up some of them take up carbon dioxide. They are autotrophic. They do not depend on the other organic or inorganic material available in the environment. They are known as autotrophs. And some of them, they cannot assimilate carbon dioxide. They have to depend on the organic and inorganic compounds available in the environment. They are known as heterotrophs. Now there are three categories on the basis of which the whole bacterial world can be divided into 6 subgroups. What are those? The carbon sources, the energy source, the electron source. Now, these three are the main components of the metabolism of any bacterial cell. Now, based on the carbon source, bacteria can be divided into autotrophs and heterotrophs, which I have already explained. The autotrophs can assimilate carbon dioxide and heterotrophs they cannot. That is why they have to depend on the organic molecules present in the environment. Now, the next one is the energy source. Based on the energy source, the bacteria can be divided into 2 groups, that is phototrophs and chemotrophs. Phototrophs are taking the energy from the photon, that is the light, and the chemotrophs they obtain their energy from chemical compounds which are present outside the cell, that is in the environment. Then comes the electron source. The electron source, based on the electron source, the bacteria can be again 2 types. First one is lithotrophs, which require the inorganic substances, and the second one is organotrophs, which requires the organic compounds. Now, I would just say that it is a very vast topic. So, it is the very basic of the bacterial nutrition and the classification of bacteria based on their nutrition. Now, this is another classification which the scientist Paul uh, described in the year of 1872. Bacteria can be divided into 3 subgroups, 3 major subgroups. First one is spherical, then rod-shaped, then spiral. Now, back in 1872, there was no infrastructures as today. That is why the shape and the structure was the criteria. Those are the morphological criteria for the basis of classification of bacteria. Now, the spherical shaped bacteria are also known as cocci. Cocci is plural and coccus is singular. Then rod shaped are known as bacilli, and then spirals. Now, the spherical shaped can be of several types, namely diplococci, then streptococci, then tetrad, then sarcina, then staphylococci. Now, if there are 2 bacterial cells together, always together, then they are known as diplococci. Then, if there are several other bacteria together, like beads in a string, they are known as streptococci. 4 together known as tetrad, 8 together sarcina, and then many together in a plane is known as staphylococci. Now coming to the rods, same, they can be of several types, and also the rod-shaped bacteria and the spirals they have flagella. Now I will discuss about cellular structure a bit after some slides. Then the spirals are known as vibrio or spirilla, and they can be small, they can be large, they can be having flagella, they can be having more than one flagella, they can be having one flagella, they can be having flagella all over their body, all over their cell wall, or they can be having the flagella which are located at one end of the bacteria. Okay, now these are the simplest classifications which scientists have done long back. Again, we can classify the bacterial world based on the oxygen requirement. Now, I have already talked about the nutritional requirement where the criteria were carbon source, energy source, and electron source. Now, I am going to talk about the oxygen requirement, which is a very useful technique or useful criteria to classify the bacteria. Now, if you take culture tubes, those tubes are actually culture tubes. If you take culture tubes, uh, yes, Those are actually culture tubes, and those yellow solutions are nothing but culture media where you can put your bacteria in. Those red color balls are representing bacteria. Now, after sometimes, like after 48 hours or after 72 hours, if you come back to your laboratory and you see the bacterial growth have accumulated, or it is concentrated into the in the upper side of the culture tube, you may know, you may confirm, you may be you can get a conclusion that your bacteria are obligate aerobes. Obligate aerobes means it cannot grow without oxygen. Okay, oxygen should be present in the environment of the cells. That is why it is known as obligate aerobes. Then you have obligate anaerobes, which will be concentrated just at the bottom of the culture tube here. Then you have, what is the meaning of obligate anaerobe? They cannot stand the presence of oxygen. That is why they are concentrated at the bottom of the test tube. Then comes the facultative anaerobe. Now, the facultative anaerobes are the bacteria which can grow in presence of oxygen, but if oxygen is not present, then also they can survive. That is the meaning of facultative anaerobe. That is the difference between obligate aerobe and facultative anaerobe. Okay, obligate aerobes are they have to have oxygen, and facultative anaerobes, if oxygen is there, they can survive. If oxygen is not there, then also they can survive. Then aerotolerant anaerobes. Aerotolerant anaerobes means they are anaerobic, but they can tolerate a little bit of oxygen. Okay, that is why they are homogeneously mixed in the culture media. Okay, now the difference between facultative anaerobe and aerotolerant anaerobe is facultative anaerobes, they prefer oxygen, but aerotolerant anaerobes, they do not have that much strong preference of oxygen. That is the difference. Then microaerophils, they prefer a little bit of oxygen. That is why they have concentrated just beneath the upper level of the culture medium. Okay. Now I would like to talk about a little bit about this bacterial cell structure. Now my colleague Dr. Fatima Calcutta has already described it. So the cells have very primitive structure. The chromosomes are naked, which has no cellular, no nuclear membrane. They have flagella, they have pili, and they have cell membrane. They have cell membrane and they have cell wall. Now, as I have told, the flagella can be uh, placed at one end. The flagella can be uh, distributed all over, and then the flagella can be at one end, placed at one end. And the first one is known as monotrichous, and the second one is known as polytrichous, and the last one is known as locotrichous. Then I would like to talk about the differences between gram-positive and gram-negative bacteria. Now, let me first tell you that bacteria, the world of bacteria can again be divided into two groups, gram-positive and gram-negative. And I hope most of you or many of you know what is gram. So, the sorry about the spelling mistake, it should be R. So the gram positive and the gram negatives is a criteria based on the gram staining procedure. Now, if you take a bacteria and if you stain them with crystal violet stain, then some of the bacteria take the stain and some of the bacteria do not take the stain. Now, I will talk about the staining. There are different types of staining of bacteria, which I will like to talk about some slides after. So, there are bacteria which can take the stain, which are known as gram positive, and there are some bacteria which do not take the stain. They are known as gram negative, and the name of the stain is crystal violet. Now, why do gram positive take the stain, and why do gram negative doesn't take the stain? Because of the cell wall structure. Okay. Now, there is a compound called peptidoglycan. Here I have written peptidoglycan is a compound which I will talk about, which is present in the gram positive cell wall in a very thick, as a very thick layer. But in the gram negative cell wall, it is very thin. This one, the middle small uh, line is representing the peptidoglycan layer in the gram negative cell wall. In the gram positive cell wall, here you can see this is the peptidoglycan layer, which is the outermost layer. Now this peptidoglycan layer takes the stain which reacts with the gram stain, that is the crystal violet. And here in the gram negative cell wall, the peptidoglycan layer is in the middle, and there is another layer, the outer membrane of the cell wall is there. That is why they cannot react with the crystal violet stain. That is why they remain unstained. We have to put another stain which is known as safranin to see them. Now under microscope you will see gram positive as blue colored cells and the gram negative as red colored cells because of the staining by counter stain safranin, not the crystal violet one. So that is another classification. Now here I have shown this one, the peptidoglycan, which I was talking about. The structure is a little complicated, but this is how it is. So there are two compounds which are known as NAM and NAC. NAM stands for N-acetyl muramic acid and NAC stands for N-acetyl glucosamine. They are joined, and the NAM is again having 4 amino acid subunits, 4 molecules of amino acids. Now this is the structure of peptidoglycan. Now I would like to talk about a little bit of reproduction, the bacterial reproduction. The bacteria are very simple organisms. They are unicellular and prokaryotes. They do not have the nuclear membrane. That is why their DNA is naked. Those are the DNA. Now, while they have to reproduce, they will just replicate their DNA into 2, and there will be cleavage between these 2 nucleoid. Here is the cleavage, starting of the cleavage. Then the cleavage will be completing, and there will be 2 complete cells. That is how it is, and this is known as binary fission. Now, it is seen that one cell is divided into 2, and this cycle will repeat on and on. Now, these 2 cells will go on replicating and will form many cells. Now, the bacterial growth curve represents how the bacterial culture is growing, or the bacterial cells are growing. Now, the replication of the reproduction of the bacterial cells cannot go on and on. I mean, infinitely, it is not possible. Why? Because the nutrients which you are giving in the culture tube is limited. So if you start with a few cells of bacteria, they will eat up the nutrient available in the culture culture media, and then after that they will start growing, they will replicate, they will exponentially replicate very fast. Then after that, what will happen? It will reach to a stationary phase. What is stationary phase? Where the nutrients are getting depleted and the cells are getting sedimented at the bottom of the culture tube. Now here you can see in this graph, along the x-axis time has been plotted and along the y-axis the number of bacteria in logarithmic term have been plotted. Now, as I have said, the bacteria will grow here exponentially and they will reach to the stationary level where it is not replicating anymore and the number of bacteria is static. Number of bacteria is not changing. Then after that, the bacterial cells will start dying. That is known as death phase. Okay. Now the brown color represents the dead bacteria and the green color represents the living bacteria. Okay. And the starting point is known as lag phase, where there is no replication. It is the you can say that is similar to stationary phase. Why it is similar? Because the bacteria which has been incorporated into the new media is getting accustomed here. It is not replicating, it is not doing anything, it is just accustomed with the new environment. This is the culture media and everything, and then it will start replicating. When it starts, it will replicate in a exponential way. Okay. Now I will like to talk about the some I would like to talk about the recombination of genetic material in bacteria. Now, the recombinations in bacteria are of three types. First is conjugation, then we will like to talk about, we will learn about the transduction and then transformation. Now, what is conjugation? It is very simple that uh, two bacterial cells will be there and there will be one bridging conjugation pilus by which two bacterial cells will be joining together. Then this is the uh, chromosome of one bacteria and this is the chromosome of another bacteria, and this is known as the plasmid. Okay. Now, when I showed you the cellular structure of bacteria, there was chromosome. Now, in almost all bacteria, they have an extracellular genetic material which is known as plasmid, okay, which is double-stranded circular genetic material, double-stranded circular DNA. Okay, now this plasmid is carrying some important characteristics, important characters like antibiotic resistance, like secretion of some proteins which are specially secreted for their defense. Now, these characteristic features, if passed from one bacteria to another, then the whole bacterial population will have the advantage of survival. Okay, usually those characters are for better survival of the bacteria. Now, here you can see that this plasmid will be uh, transferred. The one strand of the plasmid will be transferred from one bacteria to another bacteria, and there will be two copies, one in the mother cell and another in the daughter cell, or you can say the another cell. So at the beginning, the donor cell was F plus, and the recipient cell, which is receiving the genetic material, is F minus. Okay, and at the end, it is F plus. F stands for fertility, which is nothing but a character. Okay, now this is about the conjugation, which is also known as a horizontal gene transfer in bacteria. Now comes the transduction. Now transduction, you can say it is the combination of bacteria and virus. Now there are viruses which are selectively affecting or selectively invading or infecting bacteria, which are known as bacteriophages. Now these bacteriophages are the carrier of this gene transfer among bacteria. Now how it starts? There you can see that the bacteria is having the chromosome, which is the donor cell. Then one virus is there, which is having its genetic material covered into its protein coat. Then it is coming, it is ejecting its genetic material, and the genetic material here is replicated and it is translated to chop up the bacterial chromosome, bacterial DNA. Now, after that, the bacteria, the viruses will replicate. This will, these replicated genetic material will be covered by the protein coat, and some of the viruses will take some parts of the bacterial chromosomal DNA instead of its genetic material. Own genetic material is colored as red, and the bacterial DNA has been marked with blue. Now, this virus will again infect another bacteria, that is now the recipient cell, and leave the DNA or the genetic material of the donor cell, and this genetic material will be integrated into the donor cell. Okay, now this bacteria, you can say that it is a recombinant cell. It has been recombined. Why? Because another bacterial DNA has been incorporated into its own DNA. That is why it is known as recombinant bacteria or recombinant cell, you can say. Okay, now the last one is transformation, which is the easiest and simplest form of genetic recombination in bacteria. The transformation is nothing but uptaking a native DNA fragment into bacterial cell. Now, what can happen after the uptake of DNA? It can either be integrated into the bacterial genome, or it can be chopped out with some enzymes present in the bacterial cells. Okay. Another uh, possibility is there that the DNA fragment is not linear, it is a circular DNA, that is a plasmid. Now this plasmid can be uptaken by the bacterial cell, and it can be remaining as a plasmid inside the cell. That is known as stable transformation. Now this transformation, that this stable transformation has been used by the scientist for recombinant DNA technology. Okay. Now imagine you have an insulin gene. This insulin gene, the gene which is coding for the protein insulin, that gene has been incorporated into the DNA plasmid, and then this plasmid has been incorporated into a prokaryote like, let's say, E. coli. Okay. Now this E. coli will be producing lots of insulin for you. Okay. Now you can extract the insulin and you can sell it. Now this is the basis of the genetic engineering or the recombinant DNA technology, which is the foundation of modern day biotechnological applications. Okay. Now, as I started as I started with a statement that bacteria and viruses, now let's say bacteria, they cannot be seen. They cannot be seen with naked eyes. We have to have a microscope for it. Now, for the microscope, you have to stain the bacteria, otherwise you cannot see them. You have to put some coloring agent, which are known as stain, which reacts with some parts like cell wall of the bacteria, so that it becomes visible. Okay, so there are types of staining, different types of staining. First one is a simple staining, and the rest of the three are known as differential staining. Now, what is the difference between simple staining and differential staining? Simple staining is all the bacteria, whichever color, shape, size, whatever is that, they will be colored in one color, like blue. Like this uh, photograph has been taken of a microscopic field which has got bacteria which were stained by methylene blue. That is the blue colored stain. So all bacteria are looking blue. Now the next one is gram staining, where I have already told the basis of gram staining, where few bacteria are red, which are gram negative, and few bacteria are blue, which are gram positive. Now another type of staining is acid fast staining, in which the bacteria look stained bacteria looks red, and other bacteria doesn't take any color. We have to color them with a counter stain like methylene blue or so. Okay, and there is one endospore staining. Now endospore staining is done for the bacteria which forms endospore. Now what is endospore? Endospore is the structure which gives the bacteria a survival strategy. Now whenever the bacteria is not having proper environmental condition, they will form endospore and they will survive, and then again when the comfortable conditions arises, then they will sprout from the endospore. So bacillus is one of them which forms endospore. Now if you want to see your bacteria is endospore forming one or not, you will stain it like this. Okay, and it is blue in color. Okay, now when I say bacteria cannot be seen, I must say that yes, it can be seen. This is bacterial. This is a petri plate which is having a lot of bacterial colony. So the colony, one colony, let's say this one, this small dot, is millions and billions of bacteria. Okay, so one cell cannot be seen, but a bacterial colony you can count and you can see. Now if you have a water sample or soil sample or whatever, then you can understand, or you can actually calculate how many species of bacteria are there and what is the bacterial strength. So this is the procedure. Very briefly, I will discuss the procedure. What is this? This is known as serial dilution, where you serially dilute the stock solution of the bacteria. You take 1 ml dissolved in 9 ml of saline water, and then you take 1 ml, then again dissolve, so on and so forth. You reach up to here, and then from every culture tube you pour in a petri plate and see, observe after 48 hours, and you can count the colonies. Now here you can see 2 colonies are there. So likewise, you can count the colonies and multiply by the dilution factor, and you can get the number of bacteria present in the stock solution. Okay, and the types of colonies also you can find out. Now this is the basis of the quality control of every water sample, soil sample, food sample, beverage sample, everything, which are following in different industries. Now coming to virus, I have very short time. I have to finish the whole world of virus within very short time. So as the bacteria, the viruses are even smaller. Now the bacterial cell is ranging up to micrometer, and the viruses are even smaller. They cannot be seen in the compound microscope or light microscope. They are in nanometer range. Now the world of viruses can be classified in many ways. The first way the scientists choose according to their shape and sizes. They can be helical, they can be icosahedral, they can be complex structure. So helical means there will be helix of protein, and this is the genetic material of the virus. And icosahedral, they have many phases of the protein shell, and the genetic material is inside. Now the complex structure, it cannot be defined. It can be many phases. It looks like this under electron microscope, and the viruses can again be classified according to their genetic material. Now the genetic material of the viruses can be of broadly of two types, that is DNA and RNA. And again the DNA can be divided, I mean the DNA viruses can be divided into two groups, double stranded DNA and single stranded DNA. RNA is again uh, can be divided into two groups, double stranded RNA and single stranded RNA. And there are retro viruses which has single stranded RNA or double stranded DNA, which has got the enzyme which is known as reverse transcriptase. Okay. Now these are the viruses which are present in bacteria and archaea, and these are the viruses which are affecting vertebrates. Now there are, you can see that there are many viruses. Uh, those can be grouped as double-stranded DNA, single-stranded DNA, then double-stranded RNA, single-stranded RNA. It goes for the viruses which are affecting bacteria and archaea, and it goes for the viruses which are affecting vertebrates. Now there are a group of viruses which are also affecting plants. Now human beings are also affected by viruses. There are many viruses which are affecting selectively different organs. There are viruses which are affecting lungs. There are viruses which are affecting the stomach, the digestive system. There are viruses which are affecting the respiratory system, brain, and so on and so forth. Now, this is the simplest structure of virus, which has a coat or envelope, which is made up of lipid, and there is an inner capsid, which is nothing but the protein coat. This is the protein coat. This is the lipid envelope, which has different receptors, which has different protein or glycoprotein receptors. And here is the genetic material, which can be RNA, DNA, double-stranded or single-stranded. Now the bacteriophages which are infecting the bacteria, which looks like this under electron microscope, and you can see the dimension is over here 50 nanometer. Now the height should be not more than 100 nanometer. Okay, and the structure, I think you all know. The capsid is there, the viral genome is there. What is this? This is the sheath, which is again made up of protein, and these are the tail fibers, which is helping the virus to catch on, take a hold of the cellular membrane of the bacteria. Now here I would like to discuss about the bacteriophage replication. how bacteriophage replicates. Now, the bacteriophage replication is of two types, lytic cycle and lysogenic cycle. The lytic cycle is, I have already discussed in the bacterial recombination, this is nothing but transduction, okay, which I am not going to discuss again. And then the next one is known as lysogenic cycle. What is lysogenic cycle? What is the basic difference? Now you see this process of replication starts with number one, the phage is releasing its genetic material inside the bacterial cell. Now this genetic material is here. It can be integrated straight away as a prophage inside the chromosomal DNA of the bacteria. Now, when the bacteria is dividing into two, the viral genome is also dividing. Okay, it is still integrated. Okay, now this can go on and go on and go on. Now, this is the lysogenic cycle, and it may so happen that this small part of viral genome is excluded from the bacterial chromosome, and it is again present inside the bacterial cell as a separate entity. Okay, now there is no virus involved, but in the lysogenic cycle, what is the difference? The viral genome is getting integrated into the bacterial genome, but in transduction it is not getting integrated. The virus is coming, releasing its own DNA, and then it is getting packaged. It is forming thousands of viral particle, and then it is getting released in the environment. Then the thousands of viruses again infecting another bacteria, release their genome, and the cycle will repeat. Okay, so this is about the bacteriophage replication. Okay, now there, uh, there are many human viruses, of which I have chosen to discuss the HIV, the human immunodeficiency virus, which is an RNA virus. The replication is not so simple, but I have tried to simplify it. So there are proteins, you can see there are receptor proteins impregnated into the lipid coat, okay, which helps the viruses to find out with which host cell it will be attached. Now there will be a receptor in the host cell which will be present in the host cell membrane, and here at the first step the virus receptors will find the host receptor, okay, and they will form a bond so that virus is attached with the host cell, and then it will of course release the genetic material. Then the genetic material, because it is RNA, it will be transformed into DNA through an enzyme known as reverse transcriptase, and the process is known as reverse transcription, and then it will be integrated, replicated, and it will be assembled. Here this is the assembly using the host cell membrane, and it will bud out from the host cell, and it is free now to affect another host cell. Now this is uh, the life cycle of the HIV or AIDS virus in a very nutshell. Now I would like to talk about at last the SARS-CoV-2 or coronavirus or COVID-19. Okay. Now this is also an RNA virus. We all know that, and then we have a. I mean, this virus have a coat which is made up of lipid. That is why when you wash your hand, these viruses get killed. Okay, and there is a receptor which is known as ACE2 or ACE2 receptor on the membrane of the host cell, human cell. Okay, now for the receptor there is a protein which is known as S protein impregnated into the envelope, lipid envelope of the virus. Now this virus will be attached with the host cell here, and then it will be inside a vesicle type of structure which is known as fusion. Then the as the HIV virus do, this RNA genome will be replicated, and then it will be transcribed, and then it will be packaged, and then the virus will be released from the cell to infect another host cell. Okay. Now, during the replication, not one but thousands of viruses will be forming, and other organelles like Golgi complex, endoplasmic reticulum, ER stands for endoplasmic reticulum, is involved. Why? Because the RNA has to, RNA means the RNA genetic material of the virus has to go for translation. After transcription, it has to go for translation to form its own protein. For the translation to form its own protein, it has to take help of the host cellular infrastructure or host cellular machineries, that is endoplasmic reticulum and Golgi complex. They will produce their proteins and they will be released from the host cell. Okay. There you can see many other proteins are there, of which this S protein is most importance because it finds the ACE2 receptor in the human cell. Now, at last, I would like to thank all of you to listen to me and to be with me, and for your precious time, giving your precious time. And I would like to again thank our Honorable Vice Chancellor, Sir Dr. Dhruvoজ্যোতি Chattopadhyay, sir, who is standing in the middle, and our HOD, sir, Dr. Rajapal, and all the other faculty members. She is Shobana ma'am from Physics Department, she is Papia ma'am from our Chemistry Department, and there are our dear students. And we were celebrating our Science Day in early this year. We have many vibrant, colorful programs in events, poster presentation, model presentations, this and that going on in our department. And I am grateful to be a part of this. Thank you.
Thanks a lot, Dr. Ghosh. It was really a marvelous journey through the world of the bacteria and virus. Really, we have seen the, world actually. So I think that we should change the topic, that it should not be unseen. We have seen it already. So the bacteria and virus, the seen world we have to talk about. Okay. So, very, very, very thank you, Dr. Ghosh, for your, very nice talk, very enlightening talk. We have seriously, being a teacher of microbiology, I have also come to know so many things. See, there is no limit for learning. Okay, so maybe it's an introductory course, it is an introduction, but I have been introduced for so many things also. As you explained the, morphological features and how they can be classified according to different features of bacteria, it's seriously very nice. Thanks again, Dr. Ghosh, for being with us. Now, I would like to. Uh,
Rajat sir,
yes,
hello sir.
yes, yes,
There
yes,
are
yes,
some
sir.
questions in the YouTube. Some
Yes.
students have placed questions, so I want to go
Yes,
through the
yes,
question.
yes. Sure,
The
sure.
first question is asked by Rikta Dash. That is why gram positive has a thick layer and gram negative has thin layer of cell wall.
Acha, why gram positive has thick layer and gram negative has thin layer? Because nature have made so. Because my see Rita, this is natural. You may be shorter than me. You may be, your height may be more than mine. So that is natural. Naturally, they have been made so. Okay. Some bacteria they are having their cell wall where the peptidoglycan layer is much more and it is the outer covering, and for the gram negative bacteria they have another layer, and the peptidoglycan is present but it is as a very thin layer. Okay, that is why uh, I have explained that gram negative bacteria doesn't take stain and the gram positive bacteria they do take stain.
Okay, ma'am. There is another question from Rikta Dash only. That is, what is the name of the solution that is present in the test tube? I think it is about the uh, uh,
Name of the
what is the name of the solution that is present in the test tube? I think Rikta is asking about the nutrient media.
yes,
So,
yes, yes. Test tube. I have not shown anything other than culture media. Actually, in a culture tube, you may use many types of nutrient media. There are many types of nutrient media there. Commonly, we use nutrient broth. Okay, for culturing all type of bacteria, whether it is anaerobic bacteria, whether it is aerophilic, microaerophilic, so on and so forth, gram positive, gram negative, pathogenic, non-pathogenic. Okay, any kind of bacteria you just can use nutrient broth. And
okay,
in my diagrams, I have shown culture media in general.
thank you, ma'am. There is another question in the chat box. I think you have already given answer to it. So I just want to go through the question again, and if you want, you can ask more points. That what
Yes.
type of virus by structure COVID-19 is? It
What
is
type of virus?
by structure COVID-19 is. It is from Protham Khernar. The question is
What
from
type of
Protham.
structure COVID-19 is? It is complex. Complex.
Okay, ma'am. Thank you. There is no more questions in the both in the YouTube as well as chat box.
Should I go through the chat box, Dr. Shamshuka Chaudhary?
No, I think you have answered all the questions that is
Okay.
there. Okay.
Thank you so much. Thank you for your presence. Thank you for listening to me. Thank you.
Okay, so again, I'm thanking Dr. Ghosh, and may I have Mr. Ghosh, please? Mr. Gourab Ghosh, have you joined? If you have joined, kindly respond. Hello, Mr. Ghosh, are you there? Mr. Gourab
Uh,
Ghosh?
Sorry to interrupt, but
Yes.
Shomshubro, have you sent the new link to him?
Hello, SCC sir.
Okay. Yes, ma'am. I have already sent the link to Mr.
Okay.
Ghosh. So you can call him once and ask him to join over by the new link that I have sent to him.
Okay. Has he joined yet?
Mr. Ghosh, are you there? Mr. Gourab Ghosh? Maybe he has not joined. I would like to, request Dr. Ghosh, please, can you call once him?
Yes, ma'am. I think Atreyi ma'am is calling.
Okay, okay, yes. Hmm, hmm. So, the dear participants, please be with us. Maybe there is, as I told you, that this is a virtual platform and we are facing lots of problems for the net issues and the connection are not very well for some time. So please be with us until Mr. Ghosh is being online with us, and we will have our next session today, which will be delivered by Mr. Gourab Ghosh. He is coming from Mark Life Sciences Private Limited. So that also will be a very exciting talk. So be with us. We are coming very shortly.
Dr. Thakur Choudhury, hello.
Yes.
Shobshubro, can you please send the link again? Can you please send him the link again because he is saying that he has not received any link. Can I send my link to him?
Yes, you surely
Okay.
can do
Okay.
that.
Yes, Mr. Ghosh, are you there?
Yes, I'm there. Can you able to hear me?
Yes, we can hear you. Can you please put your laptop a bit lower so that we can see your face properly?
Is it fine now?
A bit more.
Is it
Yes,
fine?
yes, yes. So, may I request our head of the department, Dr. Rajat Pal, to please.
Yes, yes. Welcome, Mr. Ghosh, to the webinar series. On behalf of the Department of Microbiology and Biotechnology, Sister Nivedita University, I am welcoming you in this webinar. I hope that it will be a very nice session and your talk will enlighten us a lot. So, to the participant, I am introducing Mr. Gourab Ghosh. He is the Mr. Gourab Ghosh. He is the product manager of Mark Life Sciences Private Limited. Today, he is going to talk about the science behind the pure water. As you all know, that water, it's a, synonym of life, right? Without water, nothing can exist in the world. No life can exist. So water is a very important component of life and the all living system. So the pure water, the purification of water is also a very important process in our society. So Mr. Gourab Ghosh is going to talk about today how the water purity can be measured and how the water purification can be done. So everything, all the sciences behind the pure water, he is going to talk about, and we are going to learn, we are going to listen to him. So I'm inviting once again Mr. Gourab Ghosh to the session, and the screen is yours. As I'm telling to everyone that we cannot say that the stage is yours as it's a virtual platform. So I must say that the screen is yours, sir. So just have your talk, please, Mr. Ghosh.
Yeah, thank you. Thank you for all the participants, first of all, and thank you specifically for the Sister Nivedita University for hosting such a kind of conference, which is definitely going to help the students as well as the future researcher going forward. So I just like to take you some of the brief about the science behind water, because sir has already set the tone like why the water is important. So I'll just take you through why water is so much important for our regular life, as well as specifically since we are working in the lab. So how actually we can say this is very much important, and if we are not taking care of our water properly, what are the subsequent consequences going forward in the future, specifically whenever we are working in the laboratory. So my topic of discussion for today is science behind pure water. So here I just put a mask because we all are going through a very different situation. So now this is something which become a new normal of our life, because I hardly see the kind of webinar and a lot of web conference was happening earlier days before this COVID. So the before COVID and after COVID, we can really see a huge transition. So in other way to take it in a positive way, of course, we know that the COVID is impacting us in various different factors. So somehow our movement is restricted, and a lot of our nearer one and dearer one is also getting affected because of the COVID. And none of us is actually looking for it. We don't want to get rid of those particular situations. But we also are getting a very good learning lesson from the COVID. So why it is the learning lesson? So just very nice example, I could quote it. So earlier days, hardly if you'll ask any one of us, we were very much acquainted with any kind of digital platform itself. So now we are already very well equipped with that, and already the system Nivedita University is doing a fantastic job in terms of hosting such a kind of big webinar where all the students can directly participate. But just looking at the few days back, so if they would have also even hosted, the kind of participation, the kind of response they would have get it, probably not the way where we are getting it in digitally. So now directly we are operating from home majorly, and sometimes we are going to our offices for certain very critical work. It's only a digital medium which can actually help us to learn more. So this is a very basic learning for us, and which is definitely going to take us much more better future way. So in this note, so by saying that, I like to introduce myself. So once again, sir has given the input about me, what I am doing. So currently I'm working with Merck, so as a product manager for our Miliq division. So Miliq is basically the pioneer in terms of this water purification solution perspective. And in my previous background, so I did my MSc in biotechnology, and also I was in research for few years. So after that, I moved to corporate, and now I am handling the entire portfolio for this water purification chain. If you have any question related to the water, please feel free to post it in the chat box. I'll be happy to help you specifically for that. So who we are, just a very small and quick introduction about our company. We are a company named Merck. So we are a 350 years old company, and we believe the science is very much colorful and the vibrant. So earlier days we used to talk always the science in a very black and white manner. But if you really look at, science was colorful. Probably nowadays every day we are discovering this color of the science much more better way. And during our 350 of years of the journey, so we have understood that particular thing in a much more deeper aspect. So now we have 200 millions of the product to offer to all the kind of scientific laboratories wherever they are doing the research, or they are working on this certain pharmaceuticals, or they are working on some medical purposes. We have completely end-to-end solutions, and we also have various kind of division inside our mark. So we have the pharmaceutical division, and personally, from where I belongs from, we have the life science division. I am belonging from this life science division, and also we have some performance material division. If we talk about the performance materials, the very important thing over here. So nowadays we all are using this LCD and LED display, and all how it is getting manufactured. It's all getting manufactured by liquid crystals. So it's very much integral part of all kind of LED and LCD manufacturing process. And in Mark, we are very much pioneer in that segment as well. And MilliQ as a lab water solution, we are a part of this Merck Life Sciences division, and we cater to all kind of industries. So wherever there is a need of water, our MilliQ presence is there. It starts from these various research institutes, various kind of academic institute, food and beverage, automobile, pharmaceutical, diagnostic, you name any, we have the very good presence over there. So this is how this MilliQ as a division is our briefing is all about. So let's come to today's topic of discussion. So why purified water? So if we talk about the water, so the first thing is come to our mind, which is nothing but drinking water. And it's quite obvious because this is something which gives us the life. And without a good quality of the drinking water, definitely we can come across with a lot of waterborne diseases. And since all of you are very much passionate about microbiology, I'm sure that you must be knowing what are the waterborne contaminants are already there in specifically which comes from this drinking water. And that's why we need the certain level of purification while drinking. Sometimes in domestic or drinking purposes, we majorly use this RO. Earlier, we were using some of the filter, followed by UV has come to the picture. Now it's RO, UV, UF, even sometimes the TDS control. So if you talk about the TDS control, why we are talking about this TDS control? So TDS stands for total dissolved solids, which also majorly detects whatever the essential ions is required for our body that has to be very much present in our drinking water. Because without having a good quality of the drinking water, if our drinking water does not contain good amount of salts, like specifically sodium and all those things, which is very much essential for our body, definitely this is something which is going to create lot of problem associated with us going forward in future. So until unless we do have a proper concentration of this TDS, so definitely it's a point of concern. You may read a lot of papers, it's every day is coming to the news. RO drinking water is not very good. It's not very healthy. Over the period of usage, it may create a lot of problem associated with our body. But now coming to the laboratory water perspective, so do we need this drinking water in our laboratory? Can we use it? So probably not, because here we are working on a very in vitro condition, and we need all kind of contamination present in the water should be absolutely free, and we want the pH which should be absolutely 7. Anything deviation from the pH 7, probably it's not a very acceptable limit. So if it is pH 7, so that means there is a no question of presence of any kind of minerals, because minerals is something which is nothing but ions in nature that may create the problem. Now the comes thing, why do we need the good water in the laboratory? Because water is the most critical reagent in the laboratory. the moment itself we talk about water is the most critical reagent, very often whenever we work in the laboratory, so majority of the places we forget water as a reagent. We consider water as a water only. We use it for only for the some of the dissolution, sometimes for making some of the media, some of the buffer and all those things. But we forget one very critical factor. If we will not consider water as a critical reagent, definitely this is something may create a lot of problem in our experiment. So going forward, my request to all of you, please consider water as a critical reagent. So what are the things? Let's have a quick briefing about what are the water chemistry behind it, because we know this water, since we are talking about this water as a reagent, so definitely it's very much important for us to understand what is the chemistry behind water. So the chemistry starts from the nature. So once again, I'm telling you, science is very much colorful. It's depending upon what is the source of water, the water chemistry or the contamination profile of water also varies. We all know this water means H plus equals to OH minus, which is H2O. But water also contains some of the contamination along with it. And that's the reason all kind of water purification technology exists, so that we can take care of all those contamination and get various kind of purity of water based on our various application. From where the contamination comes, that comes from the source of the water. Majorly water are having two different sources. One is groundwater, another one is surface water. Groundwater is typically, you can consider the water which we majorly draw it from the borewell and all those things, that is getting considered as a groundwater. And another one is called as a surface water. So wherever the water is exposed to the nature, like pond water, river water, those are the things are called as a surface water. It's depending upon its nature, its contamination profile also varies. Groundwater, typically the ionic level is high, whereas in the surface water, probably not that level of ionic presence, but definitely we can expect very good presence of the particulate load and also various kind of presence of bacteria or the volatile organics. And we don't want any of the contamination to be present in our water whenever we are working in the laboratory, because we need absolute pure grade of water for the laboratory application. So these are the five major contamination of water. This starts from the salts, which also you can talk it in terms of these ions. Then it's come also there is some thing called organics. So organics is definitely going to create or may create a lot of problem in our experiment. Another one is the particle, and the microorganisms is something which we all know it is something may create a lot of problem in our experiment. Whenever we are working on the microbiology, if we are making some of the media preparation and buffer preparation, if our water itself contains some of the microorganisms, that may further create some of the problem. And the dissolved gases are also a problem. From where those things usually comes from, if we talk about any of the salts, I guess majorly this cation and anion, this ion can be present in any of the form. And that's why you need certain level of the filtration so that you can remove all kind of ionic contamination and get very good grade of water quality. Similar thing for the organics. So it's majorly come from various kinds of natural sources, even for the artificial sources. Natural sources definitely starting from the leaf, which contains this chlorophyll. And also if you look at the artificial sources, a lot of industrial waste is keep on directly diverting to our river. And that's why even from the government side, they are taking certain initiatives how they can restrict those things and minimize the contamination of in Ganga specifically. So that's why, like you all will know, the Clean Ganga, there is a big project is going on from the government of India perspective. And what are those contamination exactly? So this is majorly in terms of this organics. It could be various other things, and this volatile organics also very much present in the air. So this is also may create a lot of problem in our water. And similar thing, the particle which starts from the dust particle, mud particle, if anything present. So this is something is not at all we are exactly looking whenever we are working on any kind of application in the laboratory. So If we don't take care of those things, inclusive of the bacteria, which is one of the major factor for us, it may create a lot of problem in our experiment. So this is how the entire contamination. So just to summarize once again, water is having the majorly five contamination. It starts with the ions, organics, particles, bacteria, and gases. These are the five major contamination for the water. And that's why all the kind of water purification technology exists. Now let's come to what will be the possible impact if the water contains certain level of contamination. So the first things starts from repetition of the experiment. Here, very critical thing is that since water is a critical reagent and sometimes we don't consider water as a critical reagent, most of the time what happens whenever some problem will arise, will not be in a very healthy situation to understand whether the problem is coming from this water or the problem is coming from some other sources. So then we will be keep on repeating this experiment and maybe sometime anticipating the problem is not coming from the water, problem is coming from some other thing like reagents or some of the samples, anything it may be. So repetition is always a challenge. One side, you will be losing your healthy time while doing the repetition of your work. You will not be getting the good result even if you will repeat it, and at the end of the day probably your professor will question you what are you exactly doing. So you can definitely report to him or her, so sir I am keep on repeating the results but I am not keep on repeating this experiment but I am not getting a good results. So at the end of the day, results perspective, it's not. This is one. The next thing, it may damage your instrument. Since you are working on some of the sophisticated laboratories, some of the advanced level of application, so your instrument is also very costly. Just remember one thing, since all of you are already pursuing certain level of education, definitely you will be having a very big interest for moving to the college and during your college time for doing some level of your research for your own interest. If you damage the instrument, all the instrument cost very highly for any of the university. To further procure that instrument itself is a challenge because it's also linked with a lot of fundings, lot of other factors, and not only to that, once again you will be losing your time. Time is the biggest essence for today's situation, and you will be losing your samples as well. In today, we are working on this microliter or nanoliter of the sample size. If you lose that sample, what will be the consequences? Here, I'll just like to highlight one thing. Just imagine, since whomsoever is currently doing this COVID testing and all those things, if you are collecting the samples from one of the patient and diagnosing whether this patient is COVID positive or negative, if you lose that particular samples, can you go and further ask that patient, I want to take once again the your samples? So patient will not allow you. It is question of your credibility. Even if you are working in the research, it's a question of your research also. Sometimes you will not be in a position to get a very good healthy amount of the sample, which may also will be a biggest problem for you once again. And even if sometimes you will be getting some results, but it will not be well assured whatever the results you are getting, that is you are getting exactly right. So this is something is a major problem which may comes from the water. So we understand by that time what are the various contamination of water and what are the possible consequences of water. It may holds good for all kind of application. If you are working on this microbiology, so this is just an example in terms of the microbiology. So this is some plate we can see. So it's clearly seen lot of contamination is there, and this is typically came from the water whatever you used while doing the media preparation, also while diluting your samples. But ideal scenario, you should get a good specific thing where the colonies are very clear. You can easily count how many content of the bacteria is present in this specific samples. So none of us is actually looking for the result which is of this similar kind. So all of us will be looking for a very clean result. So where we can actually conclude whether this is positive, negative, false positive, false negative, anything, whatever we based on our experiment. This is a only small example in terms of whenever you are working on the microbiology. Similar thing, the molecular biology, molecular diagnostics is something was the major area of work for today. Even for this analytical application, or you name any of the application, all those places there will be a very different problem you may get it, and that all may come from the water. So if we are not actually taking care of our water quality in day-to-day basis, if we are not really monitoring it, this may create a lot of challenges. And for that, we need the proper filtration and the proper purification system in place, which will be giving us the complete visibility. And also we can understand what is the water quality we are getting, so whether it is consistent or it is giving some of the level of the fluctuation, because it does not mean any of the filtration if, or something, any of the purification system, if you put it in place today and it will work exactly the same without doing any kind of maintenance, maybe down the line 10 years there may be the chances of the fluctuation. It's almost like a car. So until unless we'll not maintain our car, so this is car is not going to give us in similar output whatever it was giving us at the time of purchase. So same thing hold good for any of the systems. So it starts from this water purification system, any analytical experiment, instrument, any biological instrument, all the places, the maintenance is something which is extremely required, and we have to have the proper system in place which can actually ensure us whatever water we are getting, so that water quality is consistent. And we should be having the real time monitoring of the quality, which can actually help us to understand if is there any fluctuation in terms of water. If it is so, we should be in a position to understand and monitor that thing as well. So these are the various kind of water purification technology exist in the world. So which starts from the distillation. We have various kind of technologies called deionization, electrodeionization, reverse osmosis, various kind of membrane filtration, ultrafiltration, activated carbon, ultraviolet. All those things are having a various different kind of mode of action, and it takes care of only certain level of the contamination, because water itself is a very vast subject. 75% of our body is full of water, 75% of our world is full of water, and similarly water also have huge level of the contamination. So let's come from the first level of the technologies. I'm sure that all of you are very well familiar with the distillation system. Even for the double distillation system, we heat it and condensed it, and finally, whatever water we are getting it, so that is comparatively the pure grade of water, which we very frequently use it for various kind of laboratory application. It's not from the college times, even from the school times. All the school, whenever we are doing the biological work, chemical works, we can definitely see lot of kind of distillation system is predominantly getting used. But over the period of time, so as a purification system, so we also understand it was having certain level of the challenges. Yes, we do use it, but still it is having certain level of challenges. Let me take you through what are the various challenges what we are usually facing from the distillation system, which starts from the quality challenges. It does not remove all kind of ions. It does not remove all the level of organics. It does not remove all the bacteria. Yes, very good amount of bacteria it can be removed, but some of the bacteria, specifically this bacterial byproduct like endotoxin, even if it can be present at the point of dispensing whatever we are drawing from the distilled water. And the biggest challenges over there was monitoring purpose. So there is no inbuilt monitoring system is there in place, which actually ensure us whatever water we are getting at the date of the installation, and down the line 3 or 4 years it is giving us the exact water quality. So quality perspective, of course, it's a challenge, because certain level of work we can definitely take it, still work it and believe it directly on the distillation. But in today's situation, we are also working on a lot of sophisticated instruments. It starts from the PCR, HPLC, and various kind of microbiological experiment also. So those places, definitely this is something which is creating the problem. Sometimes we are in a position to identify it, sometimes we are not in a position to identify the problem at all, whether it is coming from the water or it is coming from somewhere else. Scaling in terms of the maintenance, it's always being a challenge. Why it is a challenge? Because of the continuous heating, and since our water content, a lot of other factors also, it's not only the specific ion like sodium, calcium. So because of the over the period of usage, so it forms certain level of scaling. So while scaling, we need a very strong acid to use it for removal of those things. Similar to that fashion, it consumes huge level of energy. So since majorly our institutes is paying the bill, so we may not be so much bothered about how much energy consumption is there, but it's consume a very high level of electricity. And today, as a responsible citizen of India, it is also a part of our duty to save the energy. So whenever you will be working on any of the experiment, it's not specific to water purification system, you have to remember certain very important thing in mind, so that you can save certain thing and you can make the world much more sustainable. So one, in terms of savings of water, savings of electricity, because that are the two very critical factors. Until unless we are not going to do that, probably we cannot get a very good or healthy future for tomorrow. So it's really a time for all of us to shift to the clean and green technology and make the world much more cleaner and greener. Since I'm talking about the clean and green technology, how we can actually shift it and what are the technologies there with us? Start from RO. Yes, RO is having certain level of the challenges, because we know RO is also waste certain level of water. I'm sure all of you are very much familiar with the principles of RO. It's majorly the passing from the high density to low density through a semi-permeable membrane. So here we can say it's a bad contaminated water, and these places we are getting the pure water, and all those contamination is getting stagnated here in this membrane. So now the question comes, this RO and the drinking water RO, is it same? No, it's not same. The principle of working behind it, it's same. But whatever the RO we require in the laboratory, that cutoff is much more less, or rather I'll say much more higher as compared to any of the drinking water RO. Drinking water RO, typically we use maybe around zero point 0.1 micron or 0.01 micron maximum. But in the case of the laboratory, here the cutoff is something in a molecular weight basis cutoff, which is having much more better efficiency. So this RO itself is having a certain level of capacity, so that it can alone can remove more than 95% of ions, organics, particle, and bacteria. Just imagine, we need the minerals to be present in our drinking water. So definitely this RO is not exactly suitable for the drinking water. Whatever the output will come, it will not be having much of the minerals to be present. But we need the much more better capacity, so that very initial level we can take care of very good level of the contamination whenever we are working on the laboratory for the water purification purposes. Similarly, there are a lot of specialized ion exchange resins which allow us to remove various kind of ions, which starts from this anion and cation. So also we use various kind of mixed bed ion exchange resins while doing various kind of filtration, specifically for the removal of ions. Electrodeionization is one of the very unique technology which is already there in the market for the ages. So all the kind of water purification vendors majorly prefer to use this electrodeionization module. It is having a very unique principle, because if you look at this ion exchange resins over the period of time, because of the continuous usage, you have to treat these ion exchange resins and activate these ion exchange resins by using certain level of chemicals. So until unless you will not activate it over the period of time, it will not work. This electrodeionization technology, which ensures you need not to work put anything separately which is external. For example, you need not to treat those ion exchange resins externally by using any of the chemicals or any of the acids, and you need not to change these ion exchange beads also after a certain period of time. So it works in the principle there is one anode and one cathode, and alternatively place anion permeable membrane and the cation permeable membrane. So whatever water is coming over in the first, second compartment and the fourth compartment, you can clearly see all the anion is passing through the first compartment and the cation is passing through the third compartment. Same thing is happening in both these compartments, second compartment and the fifth compartment. So final water, whatever you are getting as the product water, it is majorly free from ionic contamination. So this is an extremely strong tool so that we can actually take care of various kind of ions. And activated carbon is very unique in design and unique in terms of the bits, which is there are also various kind of categories are there. Natural activated carbon, which majorly the charcoal we can say, and the synthetic activated carbon. Why it is getting used? It is getting used because for the removal of the chlorine and organics from the water, because the organics is a major problem and chlorine is nothing but ionic in nature. Similarly to that fashion, there are the UVs which is available in the market, majorly for 254 nanometer UV, which is getting used for removal of bacteria. What it happens whenever this bacteria will get exposed to this 254 nanometer UV, it will form the pyrimidine dimer, and bacteria will stop replicating. And there are also certain level of UVs which is currently very much getting used for removal of organics. Now you can definitely ask me how UV will remove organics. No, it is not directly going to remove the organics, but whenever UV will expose to the organic molecule, so organic molecule will break down to carbon dioxide and water. So there will not be any more presence of the organic molecule. We tell it as a photo-oxidation. So there are specific nanometer like 172 to 185 nanometer is getting used for the organic removal purpose. And the filtration is the main thing which actually shifted us the completely the cleaner and greener environment. There is a huge set of filtration is available in the world, which starts from the depth filter. So depth filter majorly taking care of various kind of particulate load at the very preliminary stage, and the screen filter, which is having the very defined pore size. Like here, a lot of vendors they use 0.22 micron filter at the point of dispensing. Whenever you are collecting the water, this 0.22 micron is getting used so that the final water, whatever you are getting, it is free from various kind of bacteria. So 0.22 micron is something it's very well recommended worldwide for removal for this bacteria purpose. So if you go to any of the pharmaceutical organization, any of the companies, so whomsoever is working on this microbiology, so they all are very much familiar with this term 0.22 micron filter. So it's a screen filter basically. And there are another specific filter which is called ultrafiltration. This ultrafiltration removes various nuclease as well as the endotoxin. If your working area is cell culture, sometime if your working area is various kind of PCR or RT-PCR, this ultrafiltration is something which is recommended to use. So those filter is very much standardized globally, which is also having the various kind of global validation specific data, which actually ensures those filter will take care of various kind of very good number of contaminations. So since already I told you about various kind of clean and greener technologies, so that we can take care of various kind of water contamination thing. But water itself is a very vast thing. None of the single technology can take care of all kind of contamination. So we need the combination of the technologies. So all the technologies is having some of its good pros as well as the cons. For example, ion exchange resins can take care of only ions. But what about the organs? What about the bacteria? It may further create the problem in your experiment. So that's the reason you need the combination of the technologies. So there are various kind of combination is available like that. So for example, pre-treatment pack, which initially take care of from tap water, we'll put this pre-treatment pack, which majorly to take care of various kind of particles and various kind of contamination which is coming from the water, followed by RO, which take care of 95% of the contamination, but less rest of the 5% is very good enough to create any of the problem in your experiment. And we need the absolute pure grade of water. We need the pH 7. So 5% is very much important over there. Electrodeionization module, 254 nanometer UV. Finally, up to here, you will get certain level of the purity, but it is not exactly the pH 7. You need further level of polishing. So you need the combination of activated carbon and ion exchange resin and photo oxidation UV lamp, majorly for the photo oxidation of the organic molecule, and the final water, whatever you will be getting, that is pH 7, and also we call it as a pure grade of ultra pure grade of water. Now, since I'm talking about something called pure grade of water, ultra pure grade of water, what is it all about? So it's come from various kind of global standards. So if you look at any of the global standards, so it also clearly defines waters are majorly of three types: type 3, type 2, and type 1. Type 3 water is something which we call it as a pure grade of water, which we can use it for any kind of basic laboratory purpose, like for rinsing, washing, and all those purposes. This is something can be used. Followed by type 2 grade of water, which can be used for various kind of comparatively critical application, which is also called pure grade of water for media preparation, buffer preparation, washing purposes as well. And next in ultra pure grade of water and reagent grade of water. Wherever you have any criticality application like PCR, RT-PCR, HPLC, LCMS, or anything, any sophisticated or sensitive application, you need the ultra pure grade of water for that. So that's how this is very clearly defined as per various global standards. Here I'm just giving two examples. One is ASTM, which is called American Society for Testing Materials. So they clearly define what should be the ideal water quality for type 1, similarly for type 2, and pharmacopoeia also similarly given the guideline. There are also guidelines from ISO. So all has defined what should be the ideal water quality for type 1, type 2, and type 3 grade of water. And depending upon your experiment, criticality or sensitivity of your experiment, you can decide which water will be suitable for your application. So as a MilliQ, we provide the entire range of solution for this. So please let us know. And this is how the specification looks like. Ions is 18.2 mega ohm centimeter. So this is something also called pH is exactly 7. So there are some of the theoretical algorithm is there, which is clearly said 18.2 mega ohm resistivity of the ionic contamination is equal to 1 by 18.2, which is 0.055 microsiemens per centimeter, is equal to pH 7. That means H plus equals to OH minus. There is no more presence of any kind of ionic contamination. But organics, we also define in terms of the ppb, sometimes ppm level. So ppb level talks about less than 2 microgram per liter. So this is how it is. Particulate level less than 1. Bacteria level less than 0.0 CFU per ml. So here you can say in other way 10 CFU per liter. And the pyrogen level is less than 0.001 endotoxin unit per ml. So this is how the entire specification looks like. So that's all from my side. So last but not the least, I'll say please consider water as a critical reagent, because since already you have started working in the laboratories or you are planning to work in the laboratories, so until unless you will not take care of this water part, definitely going forward you may see a lot of challenges in your experiment, and majority of the time you will not be in a position to understand whether the problem is coming from the water or somewhere else. So here I like to conclude my session over here. Firstly, my big thanks to all the participants, and the big thanks to the SNU for the encouraging the students specifically for kind of knowledge sharing initiative what you have taken. It's really very good. So definitely going forward, we can see a lot of good future scientists is coming up, who will be benefited out of your initiatives, definitely. So once again, thanks to all the faculty members from this SNU, and thanks to all the joinees. If you have any question, you can post it in the chat box. I'll be happy to answer to your queries.
Thank you very much, Mr. Ghosh. It is a immense pleasure for all of us to have such a nice talk. I mean, it's a very good thing. We have come to know so many things about the water in general. In the laboratory, we used to do is that we just take the water and we pour it. But what is actually behind what the I mean effort behind that pure water you people are doing, that we have come to know today, that how much it is difficult to make a water pure. We actually don't care about that. We just take the water and we make the media. My students are, wasting the pure water. But after this talk, I think my dear students can get to know, can understand that how important it is, how, effort is needed to make a water pure. So, the water purification is a very important part of the microbiology, and I would like to request all of my participants and my dear students not to waste water in laboratory. Okay, because see, so many people are working so hard to make the water pure for us only. So, and
Raju sir,
yes, Mrs. Sir,
there
is
are
there
some
any question?
questions. There are some
Yes,
questions
go
in
ahead.
the
Yes,
YouTube also.
yes.
Question is from Prem Khernath. The question is, sir, what are pyrogens?
Okay, so pyrogens is something which is once this bacteria will get dead, so it also produce some of the endotoxins. So this is majorly you can call it as a pyrogen. If you are working on the cell culture specifically, so pyrogen is something which can also create some of the problem in your cell line growth. For example, it will actually stop the cell to further grow. So this is how the pyrogen can be impacted in your experiment. Hope I could have able to clarify your doubts.
Okay, sir, thank you. We have another question. It is also from Prem Khernath. Sir, nowadays water purifiers come up with additional minerals added by machine. So does
Yes.
it fulfill our need?
Very good question. So thank you, the person who has asked the question. So here the thing is that yes, it's not always fulfilling our need. The purpose behind it, it always takes about TDS control. If you will put it by default as a TDS control, sometime you can see the equal changes is happening in your drinking water. So if you can really feel some of the changes in the drinking water taste, so definitely some of the problem is happening. So if you will continuously monitoring that particular factor, so it will be fulfilling your need up to certain extent. But without the monitoring, if you just keep it and you are assuming the fact you are drinking this RO water which is having the TDS control, so probably it is not going to suffice your need. So moment itself you will understand there is some deviation in terms of the taste. Please adjust it or call the vendor for adjusting it. So there is a real good concern over there then.
Okay, sir. Thank you. So there is another question from Shudipto Chaki. The
Yeah.
question is it commercially available drinking water free from all bacteria or microscopic living organisms?
Okay, good question. It is majorly free from the bacteria, or I would rather say majorly free from any of the bacteria which may create the problem in our body, but it's not 100% free from the bacteria. So this only ensure it is free from maybe it's like coliform or some of the specific thing which is creating some problem in our body. Once again, it's depend upon the vendor and depend upon the kind of assurance they are giving. So in today's scenario, maybe if we are driving somewhere in a highway road, we may get lot of new drinking water manufacturer who is putting this mineral water, but I'm not very sure how much actually the purification they are doing. But yes, some good vendor they really do take care of it, but only up to the things which create direct problem to our body. But if we are talking about something in the laboratory perspective, don't need any of the contamination.
Thank you, sir. Thank you very much. There are no more questions in the chat box. So
Okay.
over to you, Rajat sir.
Okay, thanks, Professor Thakur Chaudhary, for moderating the questions. And we are almost at the end of our session, today's session. This is the fourth day of our online program. We have only one day left, that is tomorrow. Tomorrow will be the last day of our online program. It is a very good experience for me. I hope all of you, whoever are joining with us, you are also sharing the same experience with me. Tomorrow again we will meet at the same time. The links of the meeting will be sent tomorrow tonight to all of you. And once again, I must thank to Professor Atreyi Ghosh and Mr. Gourab Ghosh for giving their time for their lecture and this wonderful talk. So thank you both once again, and thanks to my dear participants also for being with us for a long time. And hope to see you tomorrow again at the last day of our online program. So bye for now, and see you tomorrow again. Goodbye. Have a nice day.
Thank
Thank
you. Thanks all of you.
Yes.